I have a PhD. I ended up teaching at the University of Wisconsin School of Medicine. And then later I ended up at Stanford University School of Medicine, where my final research, I did it and I said, I got the system, I got to get out. Okay. What was I doing? I was cloning stem cells. What are stem cells? Well, everybody, these are cells in our body. I'll tell you what they are. They're embryonic cells. I go, why should I have embryonic cells in me? I go, you're made out of 50 trillion cells, and every second you lose 3 million cells.
Oh, shit.
Every second we've been talking. 3 million cells dying. Yeah,
Give
run!
me them embryonic
Use your life
cells
right now before they run out. So stem cells are cells in your body that replace the ones you're losing. So that keeps us in the current state. So they're embryonic cells. Back in 1967, while you were in nappies, maybe you were not even there yet.
I was in I was in my father's testes and and held somewhere suspended in the unborn.
And to have later opportunity, you were already being programmed in that place before you got out of it.
Trauma, inherited programming.
That's a later conversation. Programming is inherited from parent to child. Okay, so I go. So what? I was cloning stem cells in 1967. There were only a handful of us in the world that even knew what the hell a stem cell was. And I was cloning them. I go, what's that? I say, I put one cell in a dish by itself, one stem cell. It divides every 10 hours about. So first there's 1, then there's 2, then there's 4, 8, 16, 32. After a week, 30,000 cells in the Petri dish. And I go, okay, I got 30,000, what? Identical cells because they came from one parent. So 30,000 genetically identical cells. I split them into 3 Petri dishes. And I grow cells in the lab in something called culture medium, a fluid, an environment. I go, what's culture medium? The laboratory version of blood. If I'm growing human cells, I look at what's human blood made out of, and then in the lab I mix the chemicals and I feed the cells in a culture dish so they feel at home. And I go, so what? I said, well, in my experiment, I changed the chemistry of the culture medium slightly. So I have 3 different dishes, all genetically identical cells, and 3 different versions of culture medium. In culture medium A, environment A, the cells form muscle. In the adjacent dish with genetically identical cells, culture medium B, the cells form bone. In a third dish, genetically identical cells, culture medium C, they form fat cells. And I stopped at this experiment and go, oh my God, I'm teaching in the medical school that genes control life. And in my tissue culture the genes didn't control any of that. Genes didn't control that.
Oh, wow.
It was the environment. And then I go, oh my God, and I tell all my colleagues, and they look at me because the big gene movement was on, and they looked at me as, well, you're a weirdo, Lipton. We all know that genes control life, and that's a stupid experiment.